The pipeline of antibiotic discovery is a major necessity. Strengthen this pipeline is a priority for human health. And the evolution of resistance definitely has impaired this ability to respond to bacterial pathogens. Um, this pipeline faces important challenges due to the lack of investment of antimicrobial research in the private sector and an economic model that discourages investment. In the last few years, however, encouraging signs are occurring, but major gaps still remain. The World Health Organization has regularly assessed the preclinical and clinical antibacterial development pipeline, and the latest report is now available in AAC. And we are going to discuss it with the lead author of this paper. Welcome to Editors in Conversation. The objectives of this podcast is to understand the process for review of the antibacterial pipeline from the World Health Organization point of view, recognize the process and gaps in antibiotic discovery, and identify the opportunities to overcome the numerous hurdles in the early stages of the antibacterial research and development space. This episode is brought to you by the Antimicrobial Agents and Chemotherapy Journal, available at asm.org/AAC. I am your host, Cesar Arias, editor-in-chief of AAC, and this podcast is supported by the American Society for Microbiology, which publishes AAC. ASA members actually get up to 50% publishing fees, and there is a very important open access now being considered by ASM that is going to be exciting for the future. Visit asm.org/membership to sign up. And don't forget to check the latest issue of AAC available at asm.org/AAC with outstanding papers on mechanism of resistance, epidemiology, pharmacology of antimicrobial agents, and clinical therapeutics, among others. We have also special collections of challenging clinical cases and stewardship papers that are available to you. Joining us, a very distinguished guest to discuss this important topic is Dr. Valeria Giganti, who is the team leader of the World Health Organization in the AMR division at Geneva, Switzerland. Valeria, welcome to the show.
Thank you, Cesar. Thank you so much for having me today.
Okay, so let's start talking about to center this discussion. I think there is an important consideration now is that the World Health Organization have what they call a bacterial priority pathogen list. Let's talk about how the World Health Organization gets to that list and who is in the list.
Yes. So, thank you, Cesar, for this question. Actually, the bacterial priority pathogen list is one of our signature products. We developed the list first in 2017. And since that time, the list has been really critical in guiding investment in R&D, but also in guiding public health activities. For instance, activities like surveillance of control of AMR. We updated this list this year in 2024, actually to reflect a little bit the evolving nature of AMR and also to incorporate the new evidence that became available. We have conducted a comprehensive assessment of drug-resistant bacterial, considering several factors such as their mortality, disability, preventability, transmissibility, resistance, but also treatment availability, including pipeline candidates. The new BBPL contains 15 families of drug-resistant pathogens, and they are clustered in 3 groups: critical, high, and medium. In the critical group, we find Acinetobacter baumannii, Enterobacteriales, and rifampicin-resistant Mycobacterium tuberculosis. In the high priority group, we found community pathogens prevalent in low and middle income countries, such as Salmonella and Shigella, that gained also a prominent place in our update. In this group, we also have Neisseria gonorrhea and MRSA. And lastly, in the medium priority category, we have bacteria such as group A and B streptococci and Streptococcus pneumoniae. So the new list overall emphasizes the need for more R&D towards the priority pathogens and also public health efforts, especially in low and middle income countries. The BBPL is one of the different projects that WHO is leading to support R&D We also assessed, as you mentioned, the antibacterial pipeline on an annual basis. And our latest 2023 report, published in June, was actually conducted already against this updated list.
Hey, Valeria. I guess VRE is also vancomycin-resistant drug oxide is also included there in the sort of high priority, and it is important because um, if pharmaceutical companies and biotech companies can direct specific efforts to this. Since the 2017 that this bacterial priority list has been put forward, what have you noticed in terms of research and targeting, and how you said is guiding efforts in drug development? Can you just elaborate a little bit more about that?
Yes, because the pipeline also offers us the chance to see how the developments towards the different priorities shift through the years. So, for instance, we have noticed that R&D is addressing more prominent the priorities that rank the highest in the list. But we also recognize that for some priorities, other types of intervention from public health point of view, for instance, immunization strategies might be more appropriate.
Yeah, so that this is a very important, and you can access that. This also actually differs a little bit with the what we call the CDC pathogens, what they call urgent pathogen. For example, the CDC considers multi-drug resistant Neisseria gonorrhea as a critical pathogen, as an urgent pathogen, whereas the World Health Organization lists that as a high pathogen. So there are differences in between these two based on geographic interest and epidemiology. Um, so
Yes, sir. And in this regard, I want to not to remark the fact that the CDC list takes into account only one region, that is the region of the America for us, while the WHO is a global list that takes into account global priorities over the 6 WHO regions. Probably this is also the difference in for which the evidence are generated to support the development of the list. And indeed, gonorrhea is a prevalent sexual transmitted infection that is also disproportionately affecting young children and women, particularly in low and middle income countries. So for us, is also an important pathogen to be addressed.
yeah, specifically, and I also want to give an important notice that the World Health Organization is also working or has worked on a fungal pathogen priority list based on resistant patterns. Can you just tell us a little bit about that too?
Thank you, Cesar, for this question that actually opens an entire different work streams around fungal pathogens. We published the first WHO fungal priority pathogen list in 2022. And this year, actually, against this list, we are revising the pipeline of antifungal agents in clinical and preclinical development. And this project is currently ongoing. And again, as part of the R&D efforts to address the fungal priorities, we are also revising the landscape and pipeline of diagnostics for invasive fungal infection. And this project is also currently ongoing at WHO as part of the different approach to tackle invasive fungal infection.
Fantastic. So let's get into the meat of the manuscript. So the manuscript is now available. It's entitled Multi-Year Analysis of the Global Preclinical Antibacterial Pipeline Trends and Gaps. So let's talk about how the WHO, the World Health Organization, comes up with this analysis. What is the methodology used? Who participates? How do you choose these participants, etc.?
Indeed, Cesar, thank you so much for the question. The pipeline of antibacterial agents in clinical and preclinical development is reviewed through a rigorous process that is led by us at the WHO Secretariat in collaboration with the pipeline team and also in collaboration with the WHO advisory group on the R&D of new antibacterial agents. This advisory group consists of highly qualified clinicians. including yourself, microbiologists, and also other experts with expertise, for instance, in R&D, PKPD, and in general on AMR. And the advisory group is consulted through the process. The 2023 clinical pipeline update is also based on the previous edition, and we started this activity in 2017, so there have been several iterations since then. The review covers new chemical entities as well as new biological entities that are in clinical and preclinical development worldwide, but without a marketing authorization anywhere in the world. And the review is also restricted to antibacterial that are intended to treat serious and systemic bacterial infection caused by the WHO priority pathogen according to our BBPL. We also review products that are intended for C. diff and H. pylori, given the clinical importance of these pathogens. The review also includes antibiotics that are authorized, repurposed, decolonization agents, and combination therapies, given that they have at least one new chemical entities. The methodology has been consolidated and refined through the years, and we also collect data for the analysis through different sources like desktop search, literature review, and information from stakeholders. For instance, for antibacterial in clinical stage, we propose a survey to drug developers through their pharmaceutical association like BIM Alliance, Bio, IFPMA, AMR Industry Alliance. And we also perform a targeted outreach if needed. We also do literature review of peer-reviewed journals in PubMed. We search clinical trial database like clinicaltrial.gov, WHO International Clinical Trial Registry, UDRA CTD, and others. We collect conference abstract and poster, and we perform also a targeted search with the national experts from regions like Japan, the Russian Federation, who might have scientific literature in the national language. For antibacterials in preclinical development, we launch every year a WHO data call, inviting preclinical developers to submit their data, and we also complement the data from the survey through desktop search. And we perform a pre-meeting survey with the advisory groups, and we discuss results through the advisory groups consultation. For traditional agents in clinical development and authorized, we also assess their activity and their innovation potential. And for us, understanding the innovation potential of novel antibacterial is a critical step, because innovative candidates have lower chances to develop cross-resistant to existing antibiotics.
So Valeria, let's talk about that in innovation, because there are several ways to see to define that. So and I know there are several opinions of how to get to that point. So what are the main factors that WHO takes into account to have a compound that is innovative?
Thank you for this question that actually reveals a lot of behind the definition of the innovation, there has been a lot of debate within our advisory group. We have currently 4 innovation criteria. So a drug candidate to be defined as innovative needs to address at least one of our 4 innovation criteria. That is new target, a new mechanism of action, no cross-resistance, and being a new chemical entities. However, we recognize that also improvement in the clinical feature can represent an added benefit for patients. So in the discussion, in the so-called target profile, we try to reflect the potential additional benefit that these candidates brings with them.
And I guess this is an important consideration. And I remember a very interesting debate and conversation about probably the chemical entities that are more successful right now, which is beta-lactams inhibitors. So at what point we consider those innovative or not, and this is a very important concept that people need to have in mind. So any comments on that?
Indeed, the pipeline is the clinical pipeline at least is populated probably by more than half by BL-BLI inhibitors. And actually, this compound has been very successful so far, but we also recognize that mechanism of drug resistance are spreading worldwide. This is why innovation also in the chemical characteristics is an added value today to try to avoid existing potentially new emerging mechanism of resistance and ensure a longer longevity of new products on the market.
Yeah, and again, we are fighting evolution here, people. So this is a very complicated topic. So let's focus now and discuss like big findings here. So one of the first finding is like the size of the antibacterial research programs or research group continue to be very small. Um, can you comment, Valeria, on what that is and why this is not getting stronger over the year?
Yes, sure. So WHO started the pipeline review, revising the clinical pipeline. But since 2019, we also want to really look at what is happening in the preclinical space. And the aim is to provide a snapshot of the early development stage to identify potentially innovative products that may move forward and eventually reach patients one day, and also to identify some trends in the broader antibacterial R&D ecosystem. So in our preclinical pipeline analysis, we identified a total of 244 potential candidates in 2023. But this figure is consistent with previous year of analysis, maintaining a range between 2017 and 2052 programs. But this data also needs to be interpreted with cautious, because we know that only 30% of the lead compounds studied in the preclinical will potentially enter clinical trials. And this is in line, in general, with the high failure rates observed in the early pharmaceutical R&D. The attrition rate is substantial. Probably we lost between 45 and 60% of the preclinical ecosystem since 2019.
So, Valeria, sorry to interrupt. This is a very important point. So, can you expand on that? Why do you think we have such a high failure rate of compounds?
Yes, actually, the preclinical stage is a very complicated moment for drug development. We start having different challenges that are related to safety aspect. For instance, we start seeing toxicity that sometimes can be unacceptable even in animal model. We might encounter quality issues, for instance, manufacturing problems, challenges that are related to the very nature of the chemical or biological compounds. And there are also some business considerations, such as the lack of profitability or insufficient funding, that can also stop the decision to bring forward a candidate.
And I want to highlight that last point in which the economical model that pharmaceutical companies are using to develop antibacterials is probably broken in terms that the end game doesn't seem very attractive for big pharma. And I think there's a big problem there. So enough interest or investment on this preclinical pipeline is very weak. So let's continue with what you were saying.
Yes, and we also saw that overall, as you just mentioned, the number of developers is very tiny. These 244 projects are supported by 141 research groups. They are like academic group, commercial and non-profit groups, as well as private or public companies. The number of researchers has been stayed relatively stable over the last 4 years of analysis, but if we look closer at this group between 2019 and 2023, we saw that we identified 269 unique developers, but of these, only 51 groups, that is the 20% approximately, have remained consistently active across the 4 years of analysis. And again, this high turnover in the number of drug developers underlines the volatility and fragility of the R&D ecosystem. The preclinical pipeline relies on micro and small entities and academic groups. And by micro companies, we really mean startups with less than 10 employees and probably less than 6 month cash flow. The majority of this group are privately funded commercial entities. They represent approximately 86% and are followed by academic institutions and non-profit organizations. Also, we saw that the number of researchers in the EMR space is very, very tiny. We have approximately 3,000 researchers, and this number is 10 times lower than the number of the researchers active in oncology. This means that the know-how and expertise to advise this very challenging project is limited. It is also predicted to decline over time. If we look where these researchers are, for instance, most of them are in Europe and in North America. And again, a plausible explanation for this is that most of the funding opportunities are in this geography where experienced researchers in R&D with know-how have been trained and settled over the last decades.
Fascinating data and really gives a picture of what's happening with antibiotic development right now. A second point that I wanted to highlight here is in the review from 2020 onwards, the composition of the pipeline by biological modality has been relatively stable, with direct acting molecules, bacteriophage-based therapies, and indirect acting small molecule being the most represented. Can you comment on this topic, Valeria?
Thank you, Cesar. It's true, despite the high turnover in research group and research program, the composition of the pipeline has been stayed relatively stable. Since 2020, we observed an increasing number of non-traditional agents. But today, this increase reached a stabilization point. Probably this is due to the maturation or consolidation of many non-traditional agents. So since the discovery of the first antibiotic in 1910, antibacterial R&D historically focused on traditional bactericidal or bacteriostatic agents. But today, half of the program in preclinical development are direct-acting small molecules that represent the largest proportion of programs. But next to small molecules, researchers are also exploring a variety of different approaches. And among non-traditional agents that are the most represented projects, accounting altogether for the 20% of all biological modality, we have indirect acting small molecule and bacteriophage-based therapies. So these two categories of non-traditional agents also present very distinct characteristics and specific challenges for drug development. So indirect acting small molecules target virulence factors or inhibit bacterial toxin. These agents are designed to complement or enhance the efficacy of standard of care treatments rather than directly killing bacteria. On the other way around, bacteriophage have been known to the scientific and medical literature for over 100 years, but their clinical use has been primarily through compassionate use on a case-by-case basis. Today, they are being studied in randomized controlled trials in add-on to standard of care of therapies. The development pathway for this add-on therapy, including both indirect acting small molecule and phage-based therapy, requires clinical trials that are designed to demonstrate superiority. In other words, to demonstrate the added benefit in terms of clinical outcome to the background therapy. But despite the encouraging focus in the preclinical setting on non-traditional approach, the way to patients is paved by several challenges, including study design, quality aspect, and also regulatory constraints.
It is fascinating to see how these non-traditional molecules are starting to get more and more traction. There is also some sort of good news that at least in the 2023 preclinical pipeline, there was an increase in the number of programs reaching the final stage of preclinical development, which we call the INDEnabling Phase, progressing to clinical trials compared to previous years. So I see this as a good sign. What do you think, Valeria?
It's true. Actually, to try to understand better the progression and the dynamics of the preclinical ecosystem, we perform several analyses. And we group the research programs according to their self-declared preclinical stage, And then we compared the results with those of the previous years. So we saw that programs in each development stage remained relatively stable between these 4 years of analysis. This means that as projects either fail or progress into clinical development, they are replaced by new programs replenishing the preclinical pipeline. And in 2023, in this year of analysis, we notice a significant increase, approximately of 25% in programs in the IND-enabling stage. This one is probably the most critical stage of drug development, where quality and toxicity should usually arise. We observed that 19 of the 62 IND programs remained in the same stage of development since the last year of review. 24 programs transitioned here from earlier stage, and we also found 19 new programs that pop up here, contributing to the observed growth. So this remarkable growth in programs getting to the IND-enabling phase is encouraging, but also we should reflect this on whether this is a sustainable trend that we will keep observing in the future or not.
Yeah, we will see what happens in the future with this. But at least, I mean, I'm taking this as a little win in this sort of kind of sea of bad news sometimes. Let's talk about mechanism of action. I am very interested in this. Although there are some targets that are interesting intracellularly, the majority of compounds that we have put out there are sort of cell envelope, either cell membrane or cell wall agents, and that makes a lot of sense because of the obvious target that is not present in eukaryotic cells. Any comment about the actual spectrum or target of these new compounds?
Yes. So overall, we saw that when we classified preclinical programs by mechanism of action, we saw that the category we generated remained relatively stable between the 2019 and 2023. And the largest proportion, that is approximately 80% of research program, is indeed represented by candidates with a direct cell membrane effect. This category actually encompasses bacteriophage, phage-based indolysine, and direct-acting peptide programs. And we noted that renewed interest from many researchers in exploring phage-based therapies to address AMR, also employing different strategies. Some group focus on a generalized treatment option that can be used across different patients, and this is the so-called phage cocktail approach. At the same time, other researchers collect well-characterized phage, creating phage banks for a single patient use. This involved screening wastewater or sewage water from a specific region, and then matching the bacterial isolate from patient with the phage libraries to determine susceptibilities. Some other groups are exploring genetic engineered phage to enhance their efficacy, modulate host range, or to improve critical attributes such as biofilm penetration. So we also think that progress with AI and machine learning could streamline phage design processes, fostering a greater efficiency in this interesting area of research.
Fantastic. And the other important finding of this review is the topic related to what we call narrow spectrum. So let's refine what a narrow spectrum antibacterial is, and the fact that they represented more than 1/3 of the 2023 preclinical pipeline is very interesting. It's a proportion that has remained roughly comparable across the years in the first analysis in 2019. So let's define narrow spectrum and let's make a comment on that.
Thank you, Cesar. In general, antibacterial agents typically have a broad spectrum of activities, but indeed, in these studies, we found a strong focus on species-specific programs targeting a single WHO priority pathogen. And this is even a further shift from narrow spectrum to species-specific programs, highlighting the highly selectivity of these agents. So pathogen-specific agents in the preclinical pipeline range from 45% in 2020 to a low of 37% in 2023. Between 2019 and 2023, narrow spectrum program consistently also target two pathogens in particular, Pseudomonas and Mycobacterium tuberculosis. However, the use of narrow-spectrum agents required an increased use of rapid diagnostic tests. In clinical trials, we need rapid diagnostics for patient stratification, enrollment in clinical trials, and at the same time, post-marketing, we use rapid diagnostic, and matching diagnostics to ensure their appropriate use in the selected patient. So narrow-spectrum agents are actually an important approach to address AMR, but it's really important to underline that we really need a companion and matching diagnostics to enable their appropriate use in patients.
Very important concept that how infectious diseases antibacterial development has to be hand in hand with diagnostics, which there's significant advancements there, and hopefully we'll see even more next year. um, the other interesting finding is about the preclinical programs by intended target. There seems to be a lot of fluctuation across the years of analysis. Can you expand on this finding of the review, Valeria, please?
Yes, and actually this year we updated the WHO bacterial priority pathogen list, and the clusters of global priority pathogens has consequently also changed. So for antibacterials targeting the critical priorities, we observed across the 4 year of analysis that the total number of products for Acinetobacter baumannii, E. coli, Klebsiella, Enterobacteriales, and Mycobacterium tuberculosis slightly declined from 2020 to increase back in 2023. We don't know if it was due, for instance, to stagnation in the research associated to COVID or to an underreporting of programs from researchers to the WHO preclinical data call. Also, carbapenem-resistant Pseudomonas aeruginosa is now classified in the high group, whilst it was previously classified as a critical pathogens. But this classification also takes into account the decrease observed in drug resistance towards Pseudomonas. But this pathogen is also ranked 6th in terms of contribution to the global burden of disease, and thus R&D effort should not decrease towards this pathogen. In our preclinical pipeline, we also saw significant efforts to develop both broad and narrow spectrum agents against Pseudomonas. And for agents in the medium group of the BBPL, we found a limited focus on agents targeting Streptococcus pneumonia, which ranked 4th in the burden of mortality. But this could also be explained by public health efforts for this pathogen that focus more on preventative immunization strategies.
It is fascinating. So this is a massive effort and an incredible amount of data that is put into this to try to be focused and specific on this major challenge. And I strongly recommend everybody to download the paper and have a read to see details. Just to conclude, Valeria, in this amount of data, what limitations can we say are in this data that can help us understand the context of these findings?
Yes, certainly there are several limitations to this review, and we also try to find some mitigation strategies for each of the identified limitations. For instance, the primary data call was done online on the WHO webpage during the first half of 2023. And it was done against the 2017 list. However, the 2024 list was under development, and we couldn't disclose the 2024 list through the WHO preclinical data call. So to mitigate some potential reporting bias, we asked sponsors to submit information on programs that are against medically important bacteria, difficult to treat bacteria that were not included in 2017 list. Also, in our analysis, the primary data are collected to an online data called, and the data are are self-declared from the institution, from the sponsor. So where possible, WHO confirmed the integrity of such data to publication, conference abstract or poster, institutional website, or through direct contact or using other information that were in the public domain. So another limitation concerns the fact that many developers may have not evaluated the activity of their candidates against a full panel of bacterial species due to the early development stage of the preclinical research, so may have not known and reporting the potential activity against some of the pathogens listed. And it's also important to know that the few larger pharmaceutical companies that are still active in the antibacterial preclinical R&D do not often disclose their preclinical pipeline until they are mature enough to approach the clinical stage. So we encountered several challenges in analyzing the preclinical space, as not all programs are disclosed, or because there is a high also turnover rate as compared to the clinical pipeline. So probably a greater transparency in the preclinical and clinical pipeline can lead to a stronger collaboration around innovative but promising projects, support a community of scientists and drug developers, and generate more interest and funding into the drug development of novel antibacterial agents.
So a lot of work to do. So I assume this is a very important work. And so how do you see the future? How do you see this report evolving in the future? How optimistic you are at this point that we are going to get better in these drug development efforts?
So I think that a lot will depend on the new funding opportunity. And as you know, 2023 is really a crucial year for us working in AMR due to all the meetings that are laid out in preparation of the high-level meeting next September, where hopefully we will see some more commitments and funding from donors and governments to advance this important research space. I think that this will be a critical moment to see if any progress will be possible in future.
And I want to highlight that meeting, which I imagine the World Health Organization will play a major role. And I hope we can push investment in this area, critical area for human health.
Indeed.
Okay. So this is I again encourage everybody to go and read the paper. This is a crucial year for us with that UN meeting for people who work in the antimicrobial resistant space. This has been a wonderful conversation. Thank you, Dr. Yuyanti, for your insights and this leading this tremendous effort that I hope it continues in the years to come. Thank you very much. This is Cesar Arias, Editor-in-Chief of AAC, signing off.